So, Jeff, you'd mentioned that in the past, your lab had been making quite a bit of RNA, right? As things were coming up and questions were being raised and folks
Hmm.
were RNA was getting a lot hotter. I'm curious to kind of get a trajectory here of some of the innovations that your lab has been involved with, right, on the scientific side for RNA, leading up to this point. First of all, where have you been particularly active in mRNA and next gen RNA,
Sure.
R&D?
Yeah. Yeah.
Prior to the launch of the center, and then also how you see that supporting some of the work, right, that you're going to be doing moving forward with the center.
Yeah. I mean, my lab has really, for a very long time, worked on stability of
Mm,
mRNAs. That's kind of what our bread and butter is. And so, when people have asked us to work with them on therapeutic RNAs, we try to integrate all of our teachings that we've learned on mRNA stability into those molecules. And that's sort of, our knee-jerk reflex is to make
mm,
sure that we design it the way we think it should be designed, which is not necessarily as intuitive as you might think, because we
mm,
know a lot more about all the various little features that control stability. And it's not as simple as just like going to, some web protocol and hitting like
mm.
optimize my RNA and poof, there it is. There's a lot more that goes into it.
Yeah.
The other side of it is the I'm really fascinated by the RNA modification space
Yeah.
because that is an area we just simply have. Like I said, we're an N of one here, where it's
Mhm.
M1 pseudo U is what everybody throws in for the most part. I mean, there are a few others,
Mhm.
but there's so many other RNA modifications out there yet to be explored
Mhm.
that could really be used for particular applications. And then lastly, the one that you know, Matt was laughing. I said I was been working on this since 1993. It's true. And back then, we were, when somebody would say, because we used to program cells with mRNA back then, we would inject mRNAs into oocytes and stuff and reprogram cells, and people would play with this idea. Could you use it as a therapy? Well, no, because it was delivery, 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 delivery.
Mhm.
Delivery is always the problem. How do
Yeah.
you get the transcript to the cell that you care about? And, back then, nobody was really even thinking about the LNP space as a way
Mhm.
to um, not only deliver an RNA, but also to optimize delivery to specific cell types as well as affect durability and efficacy. So, what's really exciting to me is to have the center right next to these researchers that are world-class LNP experts and start exploring that space together,
Mhm.
because it is true that we're not going to innovate unless we can do that.
Yeah.
So that's another place where we've had some success, on this sort of individual basis of, here's an RNA, here's an application. Let's talk to our colleagues. Do they have an
Mhm.
LNP that might work? And all of this will have to be sort of combined as things change. For example, if you start changing modifications on an mRNA, that's going to change its chemical property.
Mhm.
As so how that gets packaged into the LNP is
That's
going to be different.
huge question.
And
Yeah.
yeah, so how do you have to optimize in that space. You
Mhm.
can't just say, hey, I just discovered this novel way of modifying an mRNA that really works great, but
Mhm.
you try to package it into a generic LNP and it doesn't do anything.
Yeah.
So having those pieces together is really important.
Mhm. An integrated
Yeah,
approach for drug product
exactly.
development. Yeah. Michelle, did you have anything to add? Or you have a background in the RNA world, right? So I mean, are there innovations that you are most excited to see the center tackle in the upcoming year or two as you set out to be COO, right? To operationalize all of these all of the goals you guys have. Are you most excited about anything that you guys are going to be working on in particular? Any innovations you want to see made?
Well, I mean, we'll start with mRNAs, and Jeff's lab has made a lot of very valuable discoveries in how to increase the stability. And as
Mhm.
Jeff has often said, if stability is increased, then you're going to have expression. So this is going to be really important. There were a lot of things that I saw at the meeting in Boston last week. Was
Mm.
that last week? 2 weeks
It
ago?
feels like it. It was 2 weeks, yeah.
There
Time
are a lot
flies.
of people. I know it really does.
Seriously.
A lot of people doing work on self-amplifying RNAs.
Mhm.
So that's an intriguing area that we don't have experience in hands-on yet, but a lot of people are asking about that.
Yeah.
And that also is going to have issues with packaging because those tend to be much larger molecules. So I'm really eager to see what happens in that space. And just how mRNAs are used in areas where they haven't been used before, such as like neoantigen therapy.
Mm-hmm.
Sorry, I'm getting a cold.
Oh no.
You jinxed me.
I wonder where that came from.
Yeah. Or for delivering enzymes and base editing and CRISPR editing. So I think a lot of people are trying to switch to that. So I think that's a really important area. We're probably going to see a lot of people asking us about that.
Yeah.
But yeah, I think self-amplifying RNA is an interesting space. I know Jeff and I have talked about that before. It has its pros and cons, but it's nice because you can deliver less and potentially get more expression. But
and potentially unmodified. There's a lot of hope that you can get the RNA to just simply be unmodified and reduce the overall, because again, this whole idea of reducing the effective dose is very important. If you're stuck at like 100 micrograms of RNA, which is what Moderna's was at, it really kind of puts you at that's a lot of RNA.
Yeah.
But if you can get it, and what I've heard from some of the self-amplifying folks is that they're down to like 0.25 micrograms. So it's a pretty significant, difference. And that puts you into a new realm of where you can start mixing and matching different strands for complex, processes, whether that's like some sort of gene editing or if it is a pan viral vaccine that you want to generate. So you can have 5 or 6 different transcripts in there, to try and get the best response that you can possibly get. So those are all things that we really need to explore.
Yeah, and these explorations, I mean, I'm reading between the lines as you talk about them, and their translation to the business aspect, right? Like, this isn't just a science experiment. This isn't just doing cool stuff. It's like, these are things that we need to do in order to move the business forward. And I'm curious about that because I don't think it can be overstated that this isn't just an academic exercise, and that you two both bring to the table a bunch of industry and vendor supplier experience. You both worked in the biotech space. You've got, like I said, supply experience. So I'm just curious about what kind of mind share that industry experience you're bringing to bear as you form the center up and create the vision and make it what you intend it to be. How have those experiences that you've had in the industry, the business of biotech, informed the way that you're setting up to help other companies build and grow?
You want to start there, Michelle?
Sure. I think Jeff handles that at the macro scale, and I handle that at the micro scale, which is to say, Jeff established the center. There are a lot of players involved, not just TriLink and Hopkins, but all the individual players within these 2 different organizations. So bringing the center in existence was, that was no small feat. And then here, getting operations going at the micro scale, I think because of my experience before in a small startup and then in a larger company, it was an eye opener to me coming from grad school and a postdoc. Where you're in a very academic mindset. It's very different than when you're in a company. Like when you are trying to sell something or help people, you have to be very customer oriented. You really need to know how to keep stakeholder engagement. You need to know never to say no or never to say it with that word. You need to understand you're trying to understand what do people need, and then how can you deliver to them, and how can you describe to them what you can and can't deliver. So I think those are the skill sets that I bring from my previous experience. And I mean, I hope it pays off.
Yeah. So
Yeah,
far,
I mean,
so good, right?
yeah, I can just to add on that. I mean, I think so. I started uh, my first biotech company back in 2018, 2017,
Mhm.
and it's Tevard Biosciences, and I started that company with Harvey Lodish, and he has really been kind of he's a sort of a icon of biotech development, and I learned a lot from him. But one thing you do learn in talking to investors and talking to other players in the space, leaders in the space, is, and it goes to why this center really is important, is that you have to hit the ground running when you start a new startup. You have a very limited time. And so you have to identify a niche that you can occupy that you bring value to. And that's really where I think we can help in this arena is if you have investigators that are coming to us and they want to do this, that, or the other thing, it's fine if you're getting a publication, great. But if you actually want to spin that out to make a company, well, then what is the real value of that product? What does it bring to the table? And we can help with that narrative
Mm,
by saying, well, we understand the mRNA space and we know
yeah.
that you have lots of competition over here, but
Hmm.
you might have a singularity here that could be explored and provide value that investors would be very interested in seeing. So, um, we don't work alone in that regard. I mean, we have the tremendous support of Johns Hopkins Technical Ventures and as well as our own contacts in the field. So I anticipate that the center will provide. We do more than just science at this center. You've mentioned we're part of this alliance for mRNA medicines, and that
Hmm.
that is another aspect because it allows us to really connect people
Yeah.
with other industrial leaders, and we have over 60 to 70 members of that alliance now that we're part of, and that network is very valuable and will be valuable to our clientele as we move forward.
Hmm. Yeah. You'd mentioned something too about how you can kind of help your knowledge of the mRNA space can help folks connect their product or their research, right, to some of the overarching needs of that space. And there's
Hmm.
a lot of best practices, I think, in terms of, how you can launch a business and best leadership practices that are really modality agnostic. But I'm curious, with your background, both of your backgrounds being in this RNA world and having
Hmm.
seen the RNA world pre-COVID when no one really gave it the respect that it was due, right? Are there any RNA specific insights that you think are really important for folks to keep in mind as they are launching an RNA business? I like to kind of dig into some of the individual nuances, right, that come from modality specific learnings and running a business.
Yeah,
So,
I mean, I think what we can see from what's happened over the last 5 years, the use of mRNA in vaccines is never going to go away.
mhm,
That's never going to go away.
yeah.
That's something that, that genie's out of the bottle, and there's a lot of issues around vaccines and political things, blah blah blah, but it's not going to go away.
Hmm.
It's just too easy, works too well, and
Hmm.
not that it works too well, it works great. Um,
Yeah.
but I think where we're really seeing incredible promise is in neoantigen therapy
Yeah.
for mRNA. Again, the idea that you can design a personalized therapy for an individual with like pancreatic
Hmm.
cancer and have that, the successes that we're seeing in the last 2 years in pancreatic cancer with neoantigen approaches is really tremendous.
Hmm.
That, this was a disease where it's really a death sentence, but was leading at least to half the people in that study were still alive several years later.
Hmm.
Um, and so that's going to continue.
Yeah.
And there's a very clear path to using mRNA in the neoantigen approaches. It's not going to work. The mRNA is it's another modality in
Yeah.
our therapeutic arsenal. It's not going to work for everything,
Hmm.
but it is going to work for these other applications. And I don't think this is a crazy vision. I think you're going to see in the next 5 to 10 years sort of point of care therapeutic development, where you have major medical centers like Johns Hopkins or Sloan Kettering or Houston Methodist creating their own programs to make personalized medicines on site, where you diagnose with some rare disease or rare cancer, you go there and they help design a therapy in the house, and you get that therapy. Now, how that all gets paid for, that's a different issue. But the technology is such that we can do it as point of care, and that's going to expand. That will expand beyond the medical community, also into like agriculture, and starting to treat our food supply before some of these pathogens emerge into the human population. mRNA is a very low cost, viable approach to start, using on some of our livestock and animal products. So,
yeah, wouldn't that be amazing? Sorry, if we could decrease the use of antibiotics and animal farming, and then maybe reduce the use of pesticides.
Yeah, exactly.
Yeah.
It's something we often don't talk about a lot in biotech, is where you have certain approaches like mRNA that can cross just from, you don't necessarily have to apply it just to humans. That technology is just as viable in other animals. So, and because it's low cost, that reduces that barrier. So,
yeah. Jeff, you talked about the valley of death earlier, and you talked about the likelihood of death, even if you make it across the valley of death. You're a very uplifting man, Jeff. You're very inspiring. I'm just kidding.
There's a movie coming soon. I feel like.
I'm curious though, and I mean, you talked about some of the causes of that mortality, but I'm curious if you've seen a lot of this, like you've seen a lot of academic exercises that showed glimpses of commercial promise and opportunity, and you've seen them go north and go south. And I'm going to ask you a hard question to answer right now, and I apologize for it in advance. If you're going to put your next paycheck on a bet, right? Like
we really are going hard here.
Yeah, there's
Michelle, you want in on this too? You gonna risk not feeding your family for a week?
You're raising the bar way too high for this question. It's not that good. But it is challenging to answer. If you had to, let's say you got 6 ideas in the lab right now. Each of them have their own, own glimpse of commercial promise down the road. But you got to put your money on what's going to go wrong, like because you've seen it go wrong before. Like what's that knowledge gap or that that missing piece that so often dooms these projects early.
Yeah, well, the biggest thing that dooms almost any biotech is, efficacy versus tox, right? Like how much benefit do you get for the risk, and is that tolerable? And we see this all the time where, there's gene therapies that are going to do great things, but then we get them into the clinic and there's very little success. As everybody's said, and you've probably heard a million times in this podcast, is that it's really easy to cure a mouse. We've eliminated cancer and most genetic diseases in mice 1,000-fold. But applying academic research to the human body is much more difficult. And so getting good efficacy and low tox is what is critical. Without that, you don't get out the door. And so if I was betting, in the place where I think a lot of people have made these bets historically is always in the cancer space, because the risk to benefit ratio is so different versus other places. Prophylactic vaccines, you've got a huge risk benefit analysis that has to be done. You cannot cause harm there at all, and it has to be efficacious. And there's a lot of political aspects about that too. But in the cancer space,
oh really? I hadn't heard.
Well,
the whole influence on the vaccine space.
we
Not
could
at
go
all.
down a whole different podcast for that one if you wanted to. But
Coming soon.
coming soon to a podcast near you. But cancer, if is in betting, where the next sort of wildly successful mRNA therapy is going to be, is in these really hard to get to cancers, these recalcitrant cancers like pancreatic cancer, glioblastoma, where we can try, we can take a lot more risk there because the benefit is so high. And that allows for innovation that then can eventually allow us to do things like monogenetic, gene replacement, and which has its challenges. The challenge there is the durability of the mRNAs. The mRNA, the reason why I study mRNA is because it goes away. That is my job. I've studied mRNA stability for decades, and it goes away, and it goes away fairly quickly. And so from a protein replacement strategy, unless we improve the durability, or we improve or if it's an indication that allows for repeat dosing, then we're sort of limited there. So from a betting standpoint, if somebody had a really good cancer neoantigen treatment, that's where I would be betting.
All right.
Yeah.
You heard it here first. Anna Rose,
but these are just I provide no market advice.
Remember, there's a paycheck riding on this. So if anyone's listening who pays Jeff's bills over here, we've
Jeff, we'll put a disclosure in the show notes.
Okay,
to protect you from any advice.
Any legal responsibilities.
Yeah, right.
We're gonna need to land this airplane.
I know.
Rose, so
We'd love to keep the planes flying here,
I've got a couple more, but I don't want to give you short shrift.